Hi everyone, I'm Cécile Poulain and I'm a PhD student at Montreal University and in the lab of Sébastien Jacquemont. And today I'm going to present you my project, which is a new GWAS method to unravel CNVs associated with autism and cognitive ability. As you may know, autism is a neurodevelopmental disorder characterized by impairment in social communication and interaction, but also restricted and repetitive behaviors. This disorder often appears in the early development and has a prevalence of 2% in the general population. Autism is often referred to as an autism spectrum disorder because it's having a large clinical heterogeneity and often co-occur with many comorbidities. For example, 30% of autism patients also have an intellectual disability. Genetic factors are known to be involved in 60% of autism cases, and many causal variants have been identified. Among these genetic variants, there is copy number variants, or CNV, which are deletions or duplications of a genomic region that may encompass one or many genes. Some recurrent CNVs have been previously associated with autism and intellectual disability with a wide range of severity and symptoms. We can see on this figure the impact of some recurrent CNV from the literature on ASD and ID risk. We can notice that the effects differ greatly depending if it's a deletion or a duplication of the region. Some CNV seems to be more associated with ASD than ID and vice versa. However, we don't fully understand these differences in the effects and the biological mechanisms that are involved. In order to disentangle the effects of CNV on ASD and ID, my project is to study the impact of genes that are encompassed in the CNVs. Our objective is to perform a CNV GWAS based on the genes to investigate the effect of CNV on the diagnostic. So to perform this GWAS, here we have the CNV for cases and control, and for each gene included in a CNV, we'll perform a statistical model that will estimate the diagnosis according to the number of carriers of this gene. We will have one statistical model per gene included in a CNV, and we will apply them separately for deletions and duplications. So for this study, we have access to 6 cohorts of unselected population and 3 cohorts of autism patients. In total, we have around 460,000 individuals, and the population that has been measured for intelligence represents 250,000 individuals, which is still a large dataset. Then we also looked at the genes or CNV already known to be associated with autism or cognitive ability in the literature, and which will allow us to verify our methodology. They will be our positive controls. So now we're going to look at the results. So first, we performed our CNV GWAS for autism. So we have the different models applied for deletion and duplication, and below we have the Miami plot where each point represents the estimation results of a gene. So at the top, we have the results for the deletion and at the bottom for the duplication. So we can see that we have different significant signals and that we found in green genes known from the literature. In a second step, we have performed 2 other CNV GWAS, one for cognitive ability and another one for autism adjusted by IQ. So the latter is a sensitivity analysis to see if we have significant genes even after correcting for the effect on IQ. These 2 GWAS were performed on the 250,000 individual datasets since we need the intelligence measures. So this graph represents all significant signals for deletion among our 3 GWAS. So for example, in the blue box, we can see that we have 3 genes that were significant in our ASD GWAS, but also in our IQ GWAS, and that these 3 genes are known from the literature in the recurrent CNV list and the SPARI database. So first, we were interested in the genes that were never observed in the literature, so in the red boxes, and which are potential new candidate genes. So first, we filter these results in order to have a robust list of new candidate genes. In order to do that, we have eliminated genes that were in a CNV with other genes known from the literature to have effect on ASD or ID and which may drive the signal. So after this step, we identified 3 new deleted genes associated with ASD. After adjusting by IQ, one of these 2 genes remains significant. Then we applied the same steps for duplications. This time we identified 25 new genes associated with ASD, and 2 of those genes remain significant after adjusting by IQ. Unfortunately, we did not identify any new genes associated with IQ, probably due to the size of the dataset. At the end, we also compared all of our GWAS results. So in this figure, the x-axis is the z-score from the IQ GWAS, and the y-axis is the odor ratio from the ASD GWAS. So each symbol represents a gene cluster. The purple symbols are the genes only significant in the ASD GWAS, and the orange symbol are the genes only significant in the IQ GWAS. And then the blue symbols are the genes significant in both of the GWAS. We can see here that we have a negative but also significant Pearson correlation for the blue genes. So as the risk of autism increases, the IQ decreases. And this observation is consistent with what is known from the literature for rare variants, and it confirms once again our new GWAS method. So to conclude, our new GWAS method seems to be robust because we have found known genes from the literature, but also new genes associated with ASD. So for the next steps, we will study the biological functions of these new genes, but also explore the biological pathways and gene dosage of all the significant signals to answer our problematic. Well, thank you all for your attention, and I hope to see you at the poster session.